working on?
Oh, hi! Just can't stop staring at our amazing wiki.
Vicky, what is it about?
Well, we've been working on developing a safer way to treat oil spills.
Sounds interesting, but I'm sure that there should be some other methods of oil cleaning, right?
Yeah, you're correct. Many methods are used for removing oil from the environment. They include mechanical, manual labor, chemical dispersants, absorbents, and in situ burning. However, existing approaches are only efficient for localized spills shortly after the accident. At the same time, they are less cost-effective and hazardous to endemic flora and fauna in the long term.
Is it related in any way to biodiversity?
Absolutely! Since it disrupts the natural habitat of many endangered species, they go extinct. This is the case for the mangrove and seal population of Caspian Sea in Kazakhstan that decreased by 90% over the past century in oil-contaminated regions.
That sounds horrible. Any way to deal with this?
Well, there are new environmentally safe and efficient methods emerging, such as bioremediation.
And what exactly is it?
It's a process that mainly uses microorganisms, plants, or microbial or plant enzymes to detoxify contaminants in the soil and other environments. We specifically focus on the use of biosurfactants for it.
Can you tell me more?
Sure, biosurfactants are active compounds that are produced by bacteria and reduce surface and interfacial tension. Using these qualities, we can emulsify crude oil.
Just any biosurfactants?
Well, we aim at rhamnolipids. They can reduce the surface tension between water and oil molecules, thereby increasing the oil degradation process performed by bacterial cells.
What kind of organisms produce them?
Well, they are usually produced by bacteria Pseudomonas aeruginosa. However, these bacteria are known for their pathogenicity because they can produce virulence factors such as exotoxins, cytotoxins, elastase, and other pathogenic proteins.
Wait, and why would you use such pathogenic bacteria on already vulnerable environments?
Yeah, the thing is, we don't. We engineer another bacteria, Pseudomonas putida, in a way that makes it produce these rhamnolipids and put it under electrofermentative conditions for higher yield.
Wow, that's creative. How did you even come up with this idea?
Previously, a team from Uppsala in 2015 and Columbia U in 2016 were involved in similar projects, but with different goals. Some of them attempted to produce rhamnolipids using E. coli to degrade PAH. Others tried to express rhamnolipid production genes in Putida to repel mosquitoes. Based on the experience of previous iGEM and new Kazakhstan teams, and using the novel dual inducible duet expression system proposed by Dr. Gautam from Berkeley Lab, we wanted to explore bioremediation methods using rhamnolipids.
Alright, so let me get this straight. Your project aims to
develop a system for increased rhamnolipids yields by overexpression of NADE under electrofermentative conditions in Pseudomonas aeruginosa and Putida. We did research on rhamnolipids production done by heterologous expression of rhamnolipid genes, RHLA-B genes. After reading different works in this field and academic literature, we found that there were already experiments trying to insert those genes into the Putida.
If making putidas that produce rhamnolipid is something that was done before, what is your novelty?
Well, the rhamnolipids production rate was low and plasmids for Putida had only one cloning site, which makes it harder to construct mutants with the potential for combined expression of NAD and rhamnolipid genes. Therefore, our team came up with the novel idea to add the NAD gene that encodes NAD synthetase to produce more NAD+, NADH, and to use PRGP-DO2 plasmid that had two multiple cloning sites to insert RHLBA genes responsible for rhamnolipid production. We enhanced this new system with electrofermentation, so more rhamnolipids will be produced.
What are the steps of your project?
Step 1 is molecular detection of NAD synthetase gene and rhamnolipid genes within electrogenetic aeruginosa. Step 2 is isolation and purification of NAD-A RHLA-B using PCR and gel elution techniques. Step 3 is cloning and overexpression of NAD-A RHLA-B genes in pseudomonads. Step 4 is determination of electrochemical potentials, NAD, NADH, and rhamnolipid yields in engineered strains of pseudomonads. Step 5 is remediation of crude oil under electrofermentative conditions using an engineered model of aeruginosa, as model for Putida.
Did your team have a certain approach in implementing this idea?
Yes, our team went through 3 iterations of a design cycle that consists of research, imagine, design, building, testing, and learning steps.
Can you follow me through all of them in consecutive order?
Sure, our experts in project implementation can tell you more about that.
So what was your first cycle about?
In the first cycle, genomic DNA was extracted from P. aeruginosa, and we attempted to amplify genes of interest using polymerase chain reaction. And we decided to use the restriction enzyme cloning method to insert NAT-E and RHLA and B genes into the plasmid. After restriction digestion, gel-purified plasmid and genes were ligated, and as a result, we obtained DUO2 plasmids carrying genes NAT-E and RHLA and B. E. coli was used as an intermediate carrier of the DUO2 plasmid to multiply the amount of plasmid and concurrently test its viability.
Have you faced any issues while doing so?
We were not able to detect the RHLA and B genes in the gel because, as we assumed, PCR amplification was not successful. So, we decided that the size of the RHLA and B gene sequence might have been the cause of the difficulties in performing PCR amplification due to the high GC content in the genome of Pseudomonas, and therefore we sought to amplify RHLA and RHLB genes separately. And that's how we transitioned into the second cycle of engineering.
What parts did you develop after cycle 1?
As we were only able to visualize the NAT-E gene, we only had the NAT-E coding sequence as a basic part. Moreover, we included our primers used for NAT-E extraction into the basic parts list.
Is a function of the part or system reproducible?
We could reproduce the PCR for isolation of the NAT-E gene, which was verified by gel electrophoresis. The bands for the NAT-E gene on the gel were visualized several times with similar results.
Now you may continue to cycle 2.
In the second cycle, through research we found that RHLA and RHLB genes can be separately used. Additionally, we increased the size of the annealing part of the primers. Therefore, we used different primers to replicate the RHLA and RHLB genes. After extraction of those genes, we amplified them in PCR and visualized PCR products in gel electrophoresis. As a result, this experiment was successful, so we plan to insert those genes into DUO2 and to deliver this plasmid into Putida. However, we faced two problems during this cycle. First, we were not able to insert all 3 genes into DUO2 plasmid, and second, Putida was not able to get our engineered plasmid.
That's unfortunate. Were there any external reasons for this?
Due to restricted access into laboratories and custom clearance, we did not have time and enough reagents to solve those issues.
What parts did you develop after cycle 2?
After this cycle, we included RHLA and RHLB genes as basic parts.
Does a part or system function under only specific host or environmental conditions?
The coding sequences like RHLA, RHLB, and NAD in our system can be used for different Pseudomonas species. Moreover, the system of all genes inserted to DUO2 can be used in E. coli.
So what did you do in cycle 3?
In the third cycle, we researched how to solve our issue, and we came up with the idea of creating 3 different plasmids with NAT-E, RHLA, RHLB genes and use P. aeruginosa instead of P. putida. This way, we plan to see the viability of each part and to test the effect of NAT-E overexpression on rhamnolipid production under electrofermentative conditions.
How did you test the functionality of this system?
The quantitative measurement of biosurfactants yields, current output, emulsification index in samples of P. aeruginosa with and without NAT-E gene were used. So we were able to see more current output through chronoamperometry and cyclic photogram in the sample of P. aeruginosa with NAT-E gene, which is red line. This proved that NAT-E gene contributed to NAD overexpression, which is in turn increased current output. We also measured the biosurfactant yields that was precipitated after our samples were introduced to oil, as well as counted the emulsification index. It showed that a sample with NAT-E gene, which is C-tube, had 2 times more precipitated biosurfactants and 1.88 times higher emulsification index than in sample with wild-type P. aeruginosa, which is A-tube.
Could you tell me more about the operations through which you are planning to modify your plasmid?
Firstly, we digested both DUO2 plasmid and genes of interest with SAC1 and SAL1 enzymes, and after we ligate plasmid by mixing it with genes in one reaction tube, introducing T4 DNA ligase with its buffer.
Sounds great, but how did you verify that you successfully inserted genes of interest into your plasmid?
After inserting genes into plasmids separately, we were able to see P. aeruginosa colony growing on plate with kanamycin. Since we didn't have access to sequencing, we extracted plasmids from those colonies, performed double digestion of those plasmids with SAL1 and SAC1 restrictases, and ran them in gel electrophoresis to obtain the following. This was quite successful since we were able to visualize two bands, one for linear plasmid and second for genes of interest. Undigested DO2 plasmid was used as a reference, and obtained results can be further applied for PPTD as well.
Therefore, would you mean that you were able to prove your hypothesis?
Yes, we observed higher current output as well as higher electrochemical potential generated by P. aeruginosa, indicating higher aerobic respiratory activity caused by increased NAD synthetase production. This, in turn, increases metabolic reactions that involve NAD as an electron carrier. It can be hypothesized that such scenario will lead to faster cell growth of bacteria, thereby inducing an increased expression of RHLB and RHLA genes and resulting in high yields of desired biosurfactants.
Earlier, you said that you introduced new parts. Were there any new composite parts?
Yes, we developed our model DO2 plasmid with inserted 3 genes, although it was not reproduced in the lab.
Cool. How do you think? Would other teams be able to build upon your work and use your parts?
Sure. Our data on primer design, RHLA, RHLB, NAD-E genes extraction can be useful for future teams on creating composite plasmids which contain all genes and introducing them into PPTD for enhanced production of rhamnolipids. Also, our finding that NAD expression increases rhamnolipid yield can be used to improve industrialization and commercialization processes.
Have you used any models?
Yes, we did 3 models for this project.
Were models used to predict the behavior of your system or guide the experimental design choices?
Models were subsequently built to characterize and explain how our system works, as well as to validate our results.
What models did you use and what important results did you get?
Well, firstly, we used Michaelis-Menten equation to predict the optimal use of substrate concentration and to calculate the catalytic rate of NAD production. And there is only a need in insertion of the amount of the measured NAD synthetase concentration for this model, and we can predict the increase in rhamnolipid production by the ratio between Vmax from our engineered strain with the NAD gene and the Vmax from strain without one. This way, we hope to use this in prediction of macro-scale rhamnolipid production. Secondly, we use 3D modeling in our amino acid sequences, and we used FIRE2 program to predict the tertiary and quaternary form of our enzymes. Moreover, we believe that these 3D models can be used in understanding the enzyme regulation and catalytic activity of those enzymes. And thirdly, our team used machine learning techniques from academic literature to predict the optimal substrate for rhamnolipid production in our engineered Putida. It showed that myristic acid was optimal one to use.
I know that your team needs to make contribution to other teams, so what did you do as a contribution?
Well, we contributed to parts for other teams that worked with RHLA, RHLB, and NADE genes. We proposed our way of extracting those genes from aeruginosa and inserting them into our plasmid. Apart from the parts, we contributed the software tools called Make It Easy and Easy Parts. We created guidebooks for summer camp preparation and improving inclusivity in iGEM teams. Finally, we also developed the improved preparation of the competent cells and electroporation of Putida.
So, how can your end product be applied?
Rhamnolipids have a very wide range of applications, from pest control to cancer research. However, the majority of these applications were not feasible in Kazakhstan due to the lack of end users.
Then how can you apply it in real life?
Kazakhstan has one of the largest oil and gas reserves in the world and hence a very well-developed oil and gas industry. Therefore, we decided to explore microbial-enhanced oil recovery and bioremediation of petroleum at contaminated sites as technologies for utilizing rhamnolipids.
Do you release bacteria itself into the environment?
No, the final product of our project is rhamnolipids. After the biosurfactant is produced in the bioreactor, the bacteria are killed. Therefore, the final product contains only rhamnolipids, which can already be used for in situ and ex situ bioremediation depending on the client's request.
But it's only theoretical, right?
The proposed model has been tested and can be used in real situations based on the results obtained. However, we yet have to figure out the way of upscaling the pathway for bioprocessing on an industrial scale.
How do you know if it's feasible on an industrial level?
Oh, we need to tell you more about our communication with the experts here.
Sounds good. I have heard that you guys have done a lot to integrate your project into the real world.
Exactly. Our approach was centered around our end users, companies specializing in bioremediation and oil spill response. Apart from that, we constantly consulted with the government representatives, science community, and ecologists to elaborate on every aspect of the project.
Did your project change as you met with more and more companies and stakeholders?
Yeah, it did. Communicating with government agencies helped to shape our understanding of the legislative part of the issue and role of bioremediation method in oil spill response in Kazakhstan. After analyzing the problems faced by our end users such as Kazakh Solutions and KMG, we realized the need for an additive product that will facilitate the action of biological methods. So, we decided to test our ramnolipids as a supplementary tool. Enhancing the ramnolipid yield and having low cost were critical questions for the commercialization prospects of our method. With the feedback from bioremediation experts, we created our model, focusing on ex situ bioremediation and paying close attention to safety.
Thanks. Now I would like to ask you about social impacts that you brought.
Oh, don't get us started here. This year, our team focused on 3 aspects: sustainability, education, and inclusivity, and also has done immense work in science communication.
Can you tell me more about your educational projects?
Happily, with the intention to increase science literacy, popularize science and biology, and tell students about iGEM, we organized a nationwide science camp that has held in 3 languages with 45 students for 5 days. We invited 28 speakers specifically for each language group, and all of them are specialists in different areas of science. Moreover, considering the lack of synthetic biology and popular science materials in Kazakh language, our team created 5 science posters, 2 crossword puzzles, and 1 board game that were spread across all the cities in Kazakhstan in 3 languages: English, Kazakh, and Russian.
You mentioned the lack of popular science materials in your country. Did you do anything else for that?
Yes, we did. Based on the materials of summer camp, we created online courses in 3 languages available for all. Also, we wrote articles about synthetic biology and bioremediation for popular science journals and educational platforms. With the goal to encourage other students of Kazakhstan to take part in iGEM, we organized an online event with iGEM alumni and one of the most popular educational projects in Kazakhstan, Leadership KZ, to talk about opportunities in iGEM and biology.
Okay. It is really impossible to stop you now. What about inclusivity?
Seeing different barriers present in STEM, we tried to raise awareness about problems of underrepresented groups. Apart from writing about it in our social media, we held a workshop, an innovative approach to inclusion and diversity, and we invited experts from different areas that are specialized in this field, so that iGEM and general community will know more about inclusivity. Consulting with specialists, we created a journal with synthetic biology materials and tactile images in Braille language and recorded audio descriptions for people with visual impairments. Also, we had quota places for children from socially vulnerable groups during the summer camp and ensured language inclusion during the whole iGEM project.
That is impressive. Any exceptional work that you are proud of?
Actually, yes. Our team also organized an international biohackathon code-on among local and international students by solving well-formulated local challenges in natural sciences to ensure the development of bioinformatics and machine learning in Kazakhstan and the world. Participants from 15 different countries solved problems with social importance and suggested their solutions for creating inclusive wiki pages, and we wrote about them in our inclusivity guidebook for other teams.
Okay, I think I should go visit your wiki and read about your socially impactful projects. Otherwise, this would not end.
Notwithstanding strict limitations due to a global pandemic, we have managed to accomplish a substantial job. We modified an environment Pseudomonas putida using a dual-inducible system that demonstrates a high yield of ranolipid production under electrofermentative conditions. Starting as an idea, we expanded the Remi project into an impactful and effective method that can positively change the environmental situation in our country and far beyond it. We believe that Remi will open up new opportunities, taking us one step closer to a clean and safe environment.
Remi, do it! Woo!